Welcome back to the Deep Dive. Today we're doing something pretty focused. We're pulling insights straight from some specialized material.
Yeah, this time it's an anesthesiology core review.
Exactly.
Yeah.
Because, when you really need to get down to the nuts and bolts of how biology works, well, you go to the people who rely on that understanding constantly.
Makes sense. Foundational stuff.
Okay, let's unpack this. So, our topic today, the neuromuscular junction, the NMJ.
That little spot.
Right. If you've ever wondered, what actually happens in those tiny fractions of a second between your brain saying move and your muscle, actually doing it.
Yeah.
This is the place.
It really is the physical connection point.
It's that crucial bridge where the electrical signal running down a nerve finally makes the jump over to the muscle fiber itself, triggering movement. So our mission really is to map out this microscopic area. We want to track that precise dance of chemicals and structures.
So you can understand exactly how movement happens at, the most basic level.
Yeah, exactly.
And what's fascinating right off the bat, I think, is the type of connection it is. We often think of nerves as just electrical wires, right?
Sure. That's the common analogy.
But here, even though speed is absolutely critical, the actual signal transmission relies on chemistry. It's a chemical synapse.
Ah, okay. Not just electricity jumping a gap.
Nope. It uses a very specific chemical messenger, a neurotransmitter, to get the job done, to turn that thought into actual muscle action.
Okay, right. So let's follow that signal then, starting with the delivery system, the motor neuron.
The source.
The command starts way back in the neuron's main body, but the actual delivery happens down that long cable, the axon, reading towards the muscle.
That's the pathway.
And our source material really highlights that this axon is wrapped in myelin, which comes from Schwann cells.
Right, the insulation.
We hear about myelin a lot, but the why here is key. That fatty sheath allows for incredibly faster signal conduction, way faster.
It's all about minimizing delay. This system is built for speed, essentially zero latency.
Like a high-speed internet cable versus dial-up.
Pretty much. Any slowdown, even tiny ones, and our movements get clumsy, uncoordinated. Speed is vital.
Okay.
And when that super fast axon gets to where it's going, it doesn't just plug into the whole muscle. No, the axon terminal branches out.
Right.
And each little axonal process connects to one single muscle fiber. It's very precise, one-to-one communication.
Okay. One nerve-ending branch talks to one muscle cell. Got it. And if we zoom right in on that very tip, the projunctional nerve ending, what's there?
Yeah, it's like a tiny warehouse stocked and ready for action.
Okay.
It's packed with mitochondria for energy, obviously.
Yeah.
But the really critical things are the storage units and the, let's call it, access control.
Solid units?
The presynaptic vesicles, little bubbles, basically.
Right.
And they're filled with the chemical messenger, acetylcholine.
Acetylcholine.
Yeah.
Okay, that's our main player chemical. And the access control.
Those are the voltage-gated calcium channels. They're the mechanism that links the electrical signal arriving in the neuron to the chemical signal getting released.
I see. So the electricity arrives.
Exactly. The electrical pulse hits, it forces those calcium channels open. Calcium ions flood into the nerve ending.
Okay.
And that rush of calcium is the biochemical trigger. It tells those vesicles, those storage bubbles, to merge with the nerve cell membrane and release their contents, the AC.
Dump to cargo.
Precisely. And this whole cascade happens to bridge that gap, which brings us to the gap itself.
Right, the synaptic cleft.
Right.
So now we've got the messenger, AC, released and floating in the space between the nerve and the muscle.
Yeah, and it's an incredibly tiny space. The source says 30 to 50 nanometers wide.
Whoa, 30 to 50 nanometers. What's a comparison for that?
Well, think about a single strand of your DNA. That's only about 2.5 nanometers wide. So this gap is maybe, 15, 20 DNA strands across? It's minuscule. And it's this tiny gap that chemically connects the nerve ending to the muscle fiber membrane, using their basement membranes as anchors, sort of.
Okay, quick question then. If the gap is that small and the nerve signal is electrical, why bother with the chemical step at all? Why doesn't the electricity just, arc across?
That's a really good question, and it comes down to control and reliability.
Okay.
An electrical jump across that gap would be, messy, maybe unreliable, potentially going in both directions, hard to regulate.
Ah, I see.
By converting it to a chemical signal, the system gains immense control. It can amplify the signal, make sure it only goes one way, and crucially, it allows the signal to be switched off really quickly. We'll get to that cleanup part later.
Okay, so the chemical step adds precision, and that chemical is acetylcholine.
That's the one. And a little historical tidbit, our source mentions AC was actually the very first neurotransmitter ever discovered.
Really, the first one. Wow. An oldie but a goodie.
Absolutely. And it's not just for muscles, you know. HE is fundamental across the nervous system. It's heavily involved in the parasympathetic system, the rest and digest functions, and parts of the sympathetic fight or flight system too.
It does a lot.
It does. But here at the NMJ, its job is crystal clear. Take that electrical command, turn it into a chemical instruction, cross the gap, and make that muscle fiber contract.
Simple mission there.
Well, the mission is simple, the execution is complex. It all kicks off when that electrical wave, the action potential, arrives and depolarizes the nerve ending.
Depolarizes, meaning the electrical charge across the membrane flips.
Exactly. A rapid change in charge. And that change is what slams open those calcium gates we talked about. Calcium floods in,
Triggering HE release.
triggering HE release into that tiny 30-50 nanometer cleft. It diffuses across almost instantly.
Okay, here's where it gets really interesting. The receiving end, the muscle side.
Right, the post-junctional membrane of the muscle fiber, the landing pad for the HE8.
And it's not just a flat surface, is it? The source mentions these things called junctional folds.
Yes, it's all folded up, like corrugated cardboard almost.
Why? Why the folds?
Surface area. It dramatically increases the surface area available for receptors.
Ah, like cramming more landing spots into the same small zone.
Exactly. Think of it like trying to catch rain. A folded tarp catches way more than a flat sheet of the same size. It maximizes the chances of catching every single AC molecule. It builds in redundancy and efficiency.
Okay, smart design. So nestled in these folds are the receivers themselves.
Yep. The nicotinic acetylcholine receptors, and that name is important, nicotinic.
Why nicotinic specifically?
Because AC can actually bind to other types of receptors elsewhere in the body, like muscarinic receptors. But here at the neuromuscular junction, it's only the nicotinic type that matters for muscle contraction.
Got it. Nicotinic at the NMJ. And these receptors, what are they exactly?
They're basically specialized ion channels, specifically sodium channels, but they're built in a really cool way. Each receptor is made up of 5 protein subunits that arrange themselves to form a central pore or channel.
5 parts.
Right. Typically, it's 2 alpha subunits and then 1 each of beta, gamma, and delta subunits, all clustered together.
Okay, 5 pieces making a gate.
But here's the really crucial bit, the part that ensures precision. Acetylcholine can only bind to the 2 alpha subunits.
Wait, out of the 5 parts, only the 2 alpha ones have docking sites for ACE.
That's right. 5 subunits, but only 2 specific binding locations.
Okay.
And get this, it's not enough for ACER to bind to just one of those alpha sites. For the receptor to actually change shape, undergo that conformational change, and open the channel,
Let me guess.
acetylcholine must occupy both of the alpha subunit binding sites at the same time.
Wow. Okay, so it's a 2-key system.
Yeah.
You need ACER in both alpha locks simultaneously to open the gate.
Exactly. It's like a biological 2-factor authentication just to make a muscle twitch.
Why such security?
Reliability. If only one HRAC molecule was needed, maybe random fluctuations or stray molecules could accidentally trigger muscle contraction. Requiring 2 simultaneous bindings at very specific sites makes activation deliberate and prevents accidental firing. It's brilliant redundancy.
I love that. A double key and lock mechanism for suture reliable activation. Okay, so both alpha sites get an HF molecule.
The receptor protein changes shape. The central channel opens up.
And ions flow.
Ions flow. Specifically, sodium ions, Na+, rush into the muscle cell down their concentration gradient.
Okay, sodium rushes in. What does that do?
That rapid influx of positive charge drastically changes the electrical potential across the muscle fiber membrane. It depolarizes the muscle cell.
Another depolarization.
Yes, and that electrical change in the muscle cell is its own action potential. That's the signal that travels along the muscle fiber and ultimately triggers the internal machinery to contract.
Muscle movement. Message received and executed.
Message executed. But the job's still not quite done. If we connect this to the bigger picture, system efficiency.
Right, because the muscle contracted, but it can't just stay contracted forever, can it?
Exactly. That signal, the 8C binding, needs to be switched off almost immediately. If the 8C just hung around in the cleft, bound to the receptors...
The muscle would stay locked, cramped.
Precisely. You'd get tetanus, paralysis in a contracted state. The system needs to reset instantly so the muscle can relax or receive the next signal, maybe milliseconds later.
So you need a rapid cleanup crew.
A very rapid and very effective one, and they need to be right there on site.
Makes sense. Who are they?
The enzyme acetylcholinesterase. A real mouthful, but crucial.
Acetylcholinesterase. Okay. And where is it?
It's strategically positioned right there in the synaptic cleft, attached to the motor end plate, basically right next door to the 8C receptors it needs to target.
Perfect location. And its job?
Its sole job is to find acetylcholine molecules and break them down. It uses water hydrolysis to split HU into two inactive pieces,
Yeah.
acetate and choline,
Signal? Terminate.
almost instantaneously. This clears the receptors, allowing the ion channel to close and the muscle fiber membrane to repolarize, readying it for another potential signal.
Efficient. But what happens to the pieces? The acetate and choline? Does the body just discard them?
No, waste not, want not. That breakdown isn't just about termination. It's actually the first step in a very neat recycling process.
Recycling. How?
The choline part specifically gets taken back up. There's an active reuptake mechanism in the presynaptic nerve terminal, the same nerve ending that released the SEH in the first place.
So the nerve ending sucks the choline back in.
Pretty much. It grabs that choline, pulls it back inside, and then uses it, along with another molecule called acetyl-CoA, which comes from the mitochondria, to synthesize brand new acetylcholine.
Yeah.
And that freshly made ACE is then pumped back into those synaptic vesicles, ready and waiting for the next nerve impulse to arrive.
So it's a closed loop. Release, bind, activate, break down, recycle, repackage.
An incredibly fast and efficient closed-loop system, built for constant, rapid repetition without running out of supplies or getting jammed up.
Okay, so what does this all mean? Let's try and summarize the whole journey for you, the listener.
Yeah, let's trace that flow one last time. It's really a cascade balancing incredible speed with absolute chemical precision.
Right. It starts with that electrical impulse flying down the neuron, sped up by the myelin sheath.
That electrical signal hits the nerve ending, triggering the chemical leap. Acetylcholine gets released and zips across that tiny 30-50 nanometer gap.
Then the receiver side, the muscle, which is physically optimized with those junctional folds, it requires that super specific double key and lock fit.
Two acerine molecules binding to the two alpha subunits of the nicotinic receptor simultaneously.
That specific binding opens the channel, sodium ions rush in, creating an electrical signal in the muscle.
Which leads to muscle contraction, movement.
And then immediately, almost as part of the same breath.
That enzymatic cleanup by acetylcholinesterase breaks down the ACE, stopping the signal.
And the choline gets recycled straight back into the nerve ending to make new AT, ready for the next round.
It's just, a marvel of biological engineering when you think about it. Balancing that speed with that specificity.
It really is stunning. The sheer precision required for even the simplest movement we make.
Absolutely. And maybe here's a final thought, something for you to chew on.
Okay.
Consider this entire cycle we just described. Release, binding, contraction, breakdown, recycling, all of that has to happen flawlessly and almost instantaneously.
Yeah.
Maybe thousands of times per second in certain muscle groups, just for you to do something fluid and seemingly simple like, say, signing your name or reacting quickly to catch something you dropped.
Wow. Thousands of times a second.
Think about the incredible speed and the incredible efficiency needed right there at that tiny junction, the NMJ, to prevent signal chaos and allow for the smooth, continuous control that defines pretty much all our physical actions. It's quite something.